Welcome to our deep dive, into the world of Rickettsia.
Oh, okay.
These are a fascinating group of bacteria, and they can cause some really serious diseases in humans. We're going to be digging into all the key details that medical microbiologists really need to know about Rickettsia.
Right.
Our main source material for this deep dive is Principles and Practice of Clinical Bacteriology, second edition by Gillespie and Hockey.
Sounds good.
Some Rickettsia are actually considered emerging infectious diseases, which is a bit concerning.
Yeah, and what makes them so interesting and also challenging is that these bacteria are what we call obligate intracellular parasites.
So they need a host cell to survive.
They absolutely depend on a host cell, not only to survive, but to replicate as well.
Interesting.
Yeah, and they have a kind of a typical bacterial morphology. They're non-motile, typically either rod-shaped or what we call cocobacillary. But the really key thing is that they reside within membrane-bound vacuoles inside the host cells.
Oh, so they're not just floating around in the cytoplasm.
Not at all. They create these little, you can think of them as almost like little hideouts or compartments.
Oh, wow.
Within the host cell where they can kind of set up shop and evade the immune system.
And that actually probably plays a role in their pathogenesis.
Exactly. And this intracellular lifestyle, it's key to understanding their pathogenesis, how they evade the immune system, how they cause disease.
Right.
And now, while we're on that subject of, related organisms, we got to mention Ehrlichia,
Okay.
another genus within the order Rickettsiales.
So closely related.
Closely related, and like Rickettsia, they're also obligate intracellular bacteria. But there's a key distinction here that's important for diagnostics.
Okay, let's hear it.
Ehrlichia lack significant amounts of peptidoglycan in their cell walls.
Peptidoglycan. So that's a major component of most bacterial cell walls, right?
Right. It provides that structural integrity and rigidity.
So what does this mean for Ehrlichia? How does this affect them?
Well, for one thing, it can affect their staining properties, which is important for us in the lab. Rickettsia, with its peptidoglycan layer, tends to stain poorly with the gram stain. Ehrlichia often won't stain at all.
Oh, interesting.
So we have to use alternative staining methods, like GIMSA or Jimenez, to actually visualize these organisms under the microscope.
So it makes them a little bit trickier to identify in the lab.
It does. And this difference in cell wall structure, it actually reflects a broader shift in how we classify these organisms.
Okay.
The order Ricketsiales has undergone some restructuring,
Interesting.
with the two main families now being Ricketsiaceae, which includes Rickettsia and Orientia, and then we have the Anaplasma, which houses Ehrlichia, Wolbachia, Anaplasma, and Neorickettsia. This reclassification, it really reflects their evolutionary relationships. It helps us better understand based on genetic analysis and molecular characterization.
It's all about those evolutionary relationships. So where do some of the other organisms that we used to think of as Rickettsia fit into this new scheme? What about organisms like Coxiella and Bartonella?
Yeah, so those have actually been moved to different orders based on, genetic and phenotypic differences. They're no longer considered part of the Rickettsiales.
So they've been kicked out of the club.
Exactly. And now, focusing specifically on the genus Rickettsia, we have three main groups that are clinically important: spotted fever group, typhus group, and scrub typhus group.
And these groups have been historically defined, I guess, based on their clinical presentations and also some serological characteristics, right?
Right. And, we used to rely heavily on those older methods like cross-immunity studies and complement fixation tests for classification. But as you can imagine, those older methods, they were often limited by cross-reactivity between different Rickettsia species, which made it difficult to get a precise identification. But thankfully, times have changed. We've entered the era of molecular techniques.
The molecular revolution.
Exactly. And with things like PCR and phylogenetic analysis, we can now identify and classify Rickettsia species with much greater accuracy and precision.
Which I imagine is critical not only for, understanding these organisms better, but also for things like epidemiological tracking and even deciding on the best treatment strategies.
Absolutely. Having that precise identification helps us make informed decisions about patient care and public health interventions.
And speaking of how these infections play out in the body, one of the reasons these infections can be so challenging is the way they target our vascular system. I mean, many Rickettsia species, they have a real affinity for endothelial cells.
Right.
Particularly those lining the small blood vessels, those tiny capillaries.
Nicely.
So they're going for the very vessels that are essential for delivering, oxygen and nutrients to tissues throughout the body.
That's right. And by damaging these endothelial cells, Ricketsiae, they essentially disrupt the integrity of the vascular system.
And that leads to?
It leads to inflammation, leakage of fluids from the blood vessels, and even clotting abnormalities. So this explains why we see such a wide range of clinical manifestations in rickettsial diseases.
So we can see anything from that classic petechial rash that we associate with some of these infections to even, more serious things like organ dysfunction.
Exactly. It can range from mild to very severe.
And it can be pretty scary, I imagine, for both the patient and the clinician.
Absolutely. One example that highlights this is Rickettsia rickettsii. This is the species responsible for Rocky Mountain spotted fever.
Right, one of the more well-known rickettsial diseases.
Exactly. And what makes this species particularly dangerous is that it doesn't just infect those capillaries, it can also invade larger blood vessels.
Oh, wow. So it's not just a matter of individual cells being infected. We're talking about more widespread vascular damage.
Precisely. And that's why Rocky Mountain spotted fever can be so severe, even life-threatening in some cases. We can see damage to multiple organs, the brain, the heart, the lungs, kidneys, you name it.
A truly systemic infection.
Absolutely. Now, it's worth noting that Ehrlichia species, even though they're also intracellular bacteria, they have a slightly different approach.
Okay.
Instead of primarily targeting those endothelial cells, they tend to infect bone marrow cells.
Interesting.
Specifically, cells of the myeloid lineage.
So these are the precursors to different types of blood cells, right?
Exactly. So infection with Ehrlichia can lead to various hematological abnormalities.
So things like leukopenia, thrombocytopenia, anemia.
Exactly. Those are all possible consequences of Ehrlichia infection. So, understanding these distinct pathogenic mechanisms, it's key to recognizing the varied clinical presentations of rickettsial and Ehrlichial diseases.
Right, because if you see a patient with those types of blood abnormalities, you might not immediately think rickettsial disease, but it's definitely something to consider.
Exactly. You got to keep it on your differential diagnosis.
Now, I think it's time to delve into those clinical presentations and really talk about, what these infections actually look like in humans.
All right, let's do it.
And, of course, we can't forget that these were arthropod-borne infections.
Right.
Meaning they're transmitted through the bites of infected ticks, lice, fleas, or mites.
Mm-hmm.
And each Rickettsia species is typically associated with a specific arthropod vector, which kind of makes sense. It influences the geographic distribution, the clinical features of the disease.
Absolutely.
It's like this ecological puzzle, where understanding the vector is this crucial piece for solving the diagnostic mystery.
You got it. So let's start with Rocky Mountain spotted fever, which we already touched on a little bit. Caused by Rickettsia rickettsii and transmitted primarily by the American dog tick and the Rocky Mountain wood tick. And this disease, it typically presents with a high fever, headache, those muscle aches, and of course, the characteristic spotted rash.
That often appears a few days after the onset of fever, right?
Right.
And it starts on the wrists and ankles.
Often, yeah. And then spreads to the trunk and extremities.
So it's a pretty distinctive rash when it's present.
It can be, but it's important to note that the rash doesn't always appear in every case.
Oh, so that can complicate diagnosis?
It can, absolutely, but when it does appear, it's a strong indicator.
Of Rocky Mountain spotted fever. Okay, so that's one. Now let's move on to another important one, epidemic typhus.
All right.
This one is caused by Rickettsia prowazekii, I believe.
That's right.
And transmitted by the human body louse.
That's correct.
And this one has a long and rather chilling history. I mean, it's often associated with war, famine, overcrowding.
It is. And historically, outbreaks have occurred in situations where you have poor hygiene and crowded conditions, which allows lice to thrive. So think refugee camps, prisons, war zones. This really underscores the link between poverty, social disruption, and infectious disease.
It's a sobering reminder that even in this day and age, these ancient diseases can really reemerge when the conditions are right.
Precisely.
Okay. Let's talk about another type of typhus.
Okay.
Marine typhus. This one is caused by, I believe, Rickettsia typhi.
Yes, Rickettsia typhi.
And transmitted by fleas, usually those that infest rats.
That's right.
And this, it tends to be a bit milder than epidemic typhus.
It is, generally, yeah.
And it's found worldwide wherever rats and their fleas are present.
It is, yeah. It has a global distribution.
So it's a good reminder to be vigilant about rodent control.
Absolutely, even in urban environments.
Even in urban environments. All right, let's talk about another one, scrub typhus. This one is caused by, Orientosutsugamushi,
That's right.
and transmitted by chigger mites.
Yes. And this one is more prevalent in Asia, Southeast Asia, and the Western Pacific. And it can present with a wide range of symptoms, from mild flu-like illness to more severe multi-organ involvement.
And I think one of the interesting things about scrubtitis is that you could sometimes see that characteristic black crusty lesion called an eschar at the site of the bite.
Yes, the eschar, it can be a valuable clue for diagnosis, but it's not always present. So clinicians need to be aware of the other clinical features of this disease.
Right. Now, there's one more rickettsial disease that I want to touch on before we move on, and that's Boutonuse fever, sometimes called Mediterranean spotted fever. This one is caused by Rickettsia connerii.
That's correct.
And transmitted by ticks.
Right.
And I think one of the things that makes this one particularly interesting is that unique black spot, that tache noire, that often appears at the site of the tick bite.
Yes, the tache noire.
So it's kind of like a little bullseye, that points right to the culprit.
Yeah, you could say that, but again, like the eschar, it's not always present.
Right, so we can't rely on it entirely for diagnosis.
No, but the disease typically presents with fever, headache, muscle aches, and a generalized rash.
So, there's definitely a lot of overlap in the clinical presentations of these different rickettsial diseases.
There is.
Which makes laboratory diagnosis all the more important.
Absolutely. And speaking of laboratory diagnosis, let's dive into how we actually identify these, often elusive pathogens.
Okay.
Now, the gold standard for diagnosing rickettsial diseases remains serology, specifically the indirect fluorescent antibody or IFA assay.
So we're looking for the presence of antibodies against Rickettsia in the patient's blood.
Exactly. We're looking for those telltale signs of the immune system's response to the infection. But while IFA is a very sensitive and specific test, it does have some limitations.
Okay, like what?
Well, one of the main challenges with serology is that it can take, a week or two for antibodies to develop to detectable levels.
Oh, so in the early stages of infection, you might not get a positive result.
Exactly. And that can delay diagnosis and treatment, which can be problematic. So while serology is a valuable tool, it's not perfect, especially when time is of the essence.
Right. And I imagine that's where molecular diagnostics like PCR are becoming increasingly important.
They are. PCR can detect Rickettsia DNA directly in clinical specimens, like blood or tissue biopsies.
So instead of waiting for the immune response to kick in and produce those antibodies, we can actually identify the bacterial culprit directly.
Precisely. And that can be a real game changer for those early stages of infection when prompt treatment is critical.
So PCR is really revolutionizing how we diagnose these infections.
It is. And there are other emerging diagnostic approaches as well, like antigen detection methods and IgM antibody capture immunoassays. These can also provide more rapid results compared to traditional serology.
So it seems like the diagnostic landscape is constantly evolving.
It is. There's a lot of exciting research happening in this area. And, while serology and molecular techniques are really the cornerstones of rickettsial diagnosis, there are other laboratory methods that can be helpful, though they're not as commonly used these days.
Okay. Like what?
So we have things like ELISA, complement fixation tests, agglutination assays, and the Weil-Felix test, which some of our listeners might remember.
Now, the Weil-Felix test, if I'm remembering correctly, it's based on this cross-reactivity between certain Rickettsia species and Proteus antigens. But it's not as reliable as some of those other methods, is it?
That's right. It's fallen out of favor due to its lower sensitivity and specificity, and also the potential for those false positive results.
Right, so not ideal. And what about culturing Rickettsia? Is that something that's routinely done for diagnosis?
Generally, no. Rickettsia species, they're notoriously difficult to grow in the lab.
Oh, really?
Right.
Why is that?
They require specialized cell culture systems and biosafety level 3 facilities. So culturing is typically reserved for research purposes or in cases where other diagnostic methods are inconclusive.
So not really practical for everyday clinical diagnosis.
Exactly. Now, let's shift gears a bit and talk about the epidemiology of rickettsial diseases. Remember that these infections are closely tied to the distribution and behavior of their arthropod vectors.
So we're talking about ticks, lice, fleas, mites. Understanding those is key.
It is. Understanding their ecology, where they live, how they behave, that's crucial for predicting where and when outbreaks might occur.
It's like creating a geographic risk map based on vector distribution.
Exactly. For example, take Rocky Mountain spotted fever. Its distribution closely follows the range of the American dog tick and the Rocky Mountain wood tick. So if you live in or travel to an area where these ticks are prevalent, your risk of encountering Rickettsia goes up significantly.
It's all about knowing your local ticks.
It is. And seasonality also plays a role. Tick-borne diseases, including Rocky Mountain spotted fever, tend to be more common during the warmer months when ticks are most active. Now, epidemic typhus, on the other hand, is a bit different. It's less dependent on those seasonal factors and more on social and environmental conditions.
So we're talking about things like overcrowded living conditions, poor sanitation, limited access to clean clothing and hygiene facilities.
Exactly. Those are the factors that drive the spread of body lice and, consequently, epidemic typhus.
It's a reminder that social determinants of health can really play a big role in infectious disease transmission.
They absolutely do. And while epidemic typhus is relatively rare in developed countries, it remains a significant public health concern in parts of the world where poverty and social disruption persist. So we can't let our guard down. Now let's talk about murine typhus, which we mentioned is transmitted by fleas that infest rats. This one has a global distribution. Wherever you find rats and their fleas, you can find murine typhus. So urban environments, especially those with poor sanitation and rodent control, they can be real hotspots for this disease.
So it's not just a rural problem. It's something we need to be mindful of even in our cities.
Exactly. And then we have scrub typhus, which we mentioned is prevalent in Asia, Southeast Asia, and the Western Pacific. This disease is often associated with rural environments, particularly areas with dense vegetation where those chigger mites thrive.
So farmers, agricultural workers, people who spend a lot of time outdoors in these regions are at increased risk.
Right. And the risk of scrub typhus can vary depending on factors like land use patterns, agricultural practices, even weather conditions. Now, shifting our focus to ehrlichiosis, again, we see that strong link between epidemiology and the distribution of tick vectors.
So similar to Rocky Mountain spotted fever, the risk of ehrlichiosis is going to be highest in areas where the tick vectors are prevalent.
Exactly. And the specific tick species that transmit ehrlichiosis can vary by geographic region. For instance, in the United States, the lone star tick is a major vector of human monocytic ehrlichiosis, or HME, and the black-legged tick is the primary vector of human granulocytic ehrlichiosis, or HGE.
So it's really important for clinicians to know which ticks are prevalent in their area, both for assessing the risk of ehrlichiosis and for guiding those prevention strategies.
Absolutely. You need to know your local ticks and the diseases they carry. Now, before we move on to those crucial aspects of prevention, treatment, and management, let's pause for a moment and test your knowledge with a little quiz.
All right, listeners, time to put those medical microbiology brains to work. Let's see how well you've been following along. So, question number 1. What's that key difference in cell wall structure that distinguishes Ehrlichia from Rickettsia? Think back to our discussion about peptidoglycan.
Question number 2. Imagine you have a patient presenting with fever, headache, and a distinctive black spot at the site of a tick bite, the tache noire. Which rickettsial disease immediately comes to mind?
Question number 3. Which laboratory diagnostic technique is considered the gold standard for rickettsial diseases? And what are some of its limitations, especially in those early stages of infection?
Question number 4. Think about those intracellular bacteria and what types of drugs can effectively target them. Which group of antibiotics is generally considered the first-line treatment for most rickettsial and ehrlichial infections?
And finally, question number 5. What are the most effective strategies for preventing tick-borne rickettsial diseases? Think about personal protection, environmental control, and the importance of prompt tick removal.
We'll review the answers and dive deeper into these crucial aspects of rickettsial disease management in the next part of our deep dive, so stay tuned.
Welcome back to our deep dive into the world of Rickettsia. Before we jump back into the clinical aspects of these fascinating bacteria, let's revisit those quiz questions. You ready to go over the answers?
I am. Let's see how our listeners did.
Okay, so question number 1, the one about that key difference in the cell wall structure between Ehrlichia and Rickettsia.
Right, that peptidoglycan issue.
Exactly. The answer is that Ehrlichia lacks significant amounts of peptidoglycan in their cell walls, unlike Rickettsia, which do have peptidoglycan. And this isn't just some, random microbiology trivia.
Oh, no, definitely not.
It has real implications.
For sure. This difference in cell wall structure affects how these bacteria stain.
Right.
So as we talked about earlier, Rickettsia, with its peptidoglycan, tends to stain poorly with a gram stain. And Ehrlichia often won't stain at all, so we have to use different staining methods in the lab.
We've got to pull out the Gimza or the Jimenez.
Exactly. Those are our go-to stains for these tricky organisms. And the lack of peptidoglycan in Ehrlichia can also impact their susceptibility to certain antibiotics.
So knowing this little difference can actually guide our treatment decisions.
Absolutely. It helps us choose the most effective antibiotics.
Okay, let's move on to question number 2.
Right?
The one with the patient presenting with fever, headache, and that distinctive black spot, the tache noire, at the site of a tick bite.
Yes, the tache noire. A real giveaway in some cases.
So which rickettsial disease are we talking about here?
That would be boot-noose fever, caused by Rickettsia connori.
Exactly. And that tache noire, while not always present in cases of boot-noose fever, it's a strong clinical sign when it does show up.
It is. It's basically a small area of necrosis where the tick bite occurred, often surrounded by a kind of reddish halo.
It's like a little bullseye.
A perfect description. All right, let's tackle question number 3. This one was about the gold standard for diagnosing rickettsial diseases.
Right, and we talked about this earlier. The gold standard is serology.
Yeah.
Specifically the indirect fluorescent antibody assay, or IFA.
Exactly. We're looking for those antibodies in the patient's blood, evidence that their immune system has encountered the Rickettsia. But, as we discussed, IFA does have its limitations.
Yeah, it's not a perfect test, especially early on in the infection when those antibodies might not have developed to detectable levels yet.
That's right. So in those early stages, clinicians really have to rely on their clinical suspicion.
Their intuition, their experience.
Exactly. And those epidemiological clues.
So has the patient traveled to an area where rickettsial diseases are common? Have they had any recent tick bites? Those kinds of...
Exactly. And this is where those molecular diagnostics, like PCR, they're really proving their worth. They offer a more rapid and sensitive way to detect the Rickettsia DNA.
And PCR doesn't rely on those antibodies. It can pick up on the bacteria directly, even in those early stages of infection.
Precisely. So it's a valuable tool for those cases where a quick diagnosis is critical. All right, on to question 4. This one was about the first-line treatment for rickettsial and ehrlichial infections.
And the answer is?
Tetracyclines, with doxycycline being the drug of choice in most cases.
Doxycycline's a good one. It can get inside those host cells and reach those Rickettsia hiding out.
That's right. And it's generally well-tolorated, relatively few side effects. But of course, with any medication, there are some things to be mindful of.
Like the fact that doxycycline can cause tooth discoloration in children.
Right, so we have to consider alternatives for those patients.
Other tetracyclines like tetracycline or minocycline can be used in adults.
Yes, and chloramphenicol is another option, though it's generally reserved for pregnant women and children because of its potential side effects. It can cause bone marrow suppression, so it's not our first choice.
It's all about weighing the risks and benefits for each individual patient.
Absolutely. And of course, the specific choice of antibiotic and the duration of treatment, that should always be individualized.
Taking into account the specific Rickettsia species involved, the severity of the illness, the patient's overall health, it's not a one-size-fits-all situation.
Exactly. Now, moving on to question 5, the one about preventing tick-borne rickettsial diseases. What are the most effective strategies?
Well, it all starts with minimizing exposure to those ticks.
Absolutely. And this involves a two-pronged approach. We need personal protection measures and we need environmental control strategies.
So on the personal protection front, we're talking about things like wearing long sleeves and pants when you're in tick-infested areas, using insect repellents that contain DT, and treating your clothing with permethrin.
All excellent strategies. And I cannot overemphasize the importance of regular tick checks after you've been outdoors.
Yeah, those little guys can be sneaky.
They can. Early detection and removal of ticks, that's crucial.
Absolutely. The sooner you get that tick off, the better.
Way.
And there's a right way to do it, too.
Oh, definitely. You want to use fine-tipped tweezers to grasp the tick as close to the skin surface as possible and pull upward with steady, even pressure. No twisting or jerking. That can increase the risk of transmitting pathogens.
Good advice. And if you're in an area where tick-borne diseases are really prevalent, it's worth talking to your doctor about prophylactic antibiotics.
Yes, especially if you've had a recent tick bite. Prophylactic antibiotics can be effective in preventing certain rickettsial diseases like Rocky Mountain spotted fever.
Yeah.
Now, in addition to those personal protection measures, there are environmental control strategies that can help reduce tick populations around your home and in your yard.
So things like keeping your lawn mowed, clearing brush, and leaf litter.
Exactly. You want to make your yard less inviting to those ticks. And we focus primarily on tick-borne rickettsial diseases, but it's important to remember those other vectors too.
Right, those pesky body lice that transmit epidemic typhus.
Exactly. Prevention, in this case, really centers around good hygiene practices, regular bathing, washing your clothes in hot water, avoiding close contact with people who might have lice infestations.
So it comes down to cleanliness and awareness.
Exactly. And with murine typhus, which is spread by fleas that live on rats, rodent control is really paramount.
So sealing up those cracks and holes in buildings, eliminating food sources, working with pest control professionals to manage those rodent populations, those are all important steps.
Right. By reducing the rodent population, you indirectly reduce the number of fleas that can transmit Rickettsia typhi. It's all about breaking that chain of transmission.
Now, before we move on to treatment and management, I want to take a little detour and explore this really fascinating topic of immunity. How does our body actually fight back against these rickettsial invaders?
It's a complex and fascinating process involving both the innate and the adaptive arms of the immune system. It's a beautifully orchestrated defense system.
So it's not just about those antibodies that we were talking about earlier. It's a whole coordinated response involving different immune cells, signaling molecules, all working together.
Precisely. So the initial response involves those innate immune cells, the first responders. These are cells like macrophages and neutrophils, and they can recognize and engulf the invading bacteria. And then they release these signaling molecules called cytokines that trigger inflammation and recruit other immune cells to the site of infection.
It's like sounding the alarm.
Exactly, calling in the troops. And as the infection progresses, the adaptive immune response kicks in, and this involves those T cells and B cells that we talked about.
So T cells, they're the ones that can directly kill infected cells, right? And B cells, they produce those antibodies that can target the Rickettsia.
That's right. Those antibodies can neutralize the bacteria, prevent them from entering new host cells, and they can also tag them for destruction by other immune cells. It's a highly effective system when it's working properly.
It's amazing how our bodies have evolved these intricate defense mechanisms.
It is. And over time, this immune response leads to the development of what we call immunological memory. So the body remembers the enemy, and it's prepared to mount a faster and more effective response next time around.
So we build up immunity. But I imagine it's not always foolproof. I mean, can you get infected with Rickettsia more than once?
That's right. Immunity to rickettsial infections isn't always complete or long-lasting. You can potentially get infected again, especially if you're exposed to a different species of Rickettsia or if your immune system is compromised in some way.
So it's not a guarantee of lifelong protection.
Unfortunately not. And this complexity of the immune response has implications for things like vaccine development.
Right, which has been a challenge for rickettsial diseases. We do have vaccines for some of them, like epidemic typhus and Rocky Mountain spotted fever, but they're not always fully protective.
Right.
And they're not available everywhere in the world.
That's right. So there's a lot of research going on in this area trying to develop more effective vaccines.
Okay, now let's shift gears and talk about treatment. We've already mentioned doxycycline as the go-to antibiotic.
Right. Early diagnosis and prompt treatment with antibiotics, they are absolutely paramount.
We can't afford to waste time with these infections.
Absolutely not. Delays in treatment can lead to more severe complications, even death in some cases. And that's why doxycycline is so important. It's a tetracycline antibiotic that has excellent activity against these intracellular bacteria. It gets right inside those cells and stops those Rickettsia in their tracks.
And doxycycline is usually pretty well tolerated by most people.
It is. But as we mentioned earlier, it can cause tooth discoloration in children, so we have to be mindful of that.
Right. So for kids, we might have to consider other options like tetracycline or minocycline or even chloramphenicol in certain situations.
Right. But again, chloramphenicol, it's not our first choice because of those potential side effects, particularly bone marrow suppression. So we have to weigh those risks and benefits very carefully.
It's a judgment call.
It is. And in addition to antibiotics, supportive care is often essential for managing those complications that can arise from rickettsial infections.
So things like intravenous fluids to maintain hydration, medications to control fever and pain, and even mechanical ventilation in severe cases of respiratory failure.
Exactly. We have to address not only the infection itself, but also those potential downstream effects.
And it's also important to remember that part of management involves educating our patients.
Oh, absolutely. Patient education is key. We need to empower our patients with the knowledge and tools they need to protect themselves and their families.
So teaching them about the risks of tick bites, how to prevent them, what to do if they find a tick embedded in their skin.
Exactly. And the importance of prompt tick removal.
Right. No procrastinating on that one.
Mm.
And we should also make sure our patients know those early signs and symptoms of rickettsial diseases so they can seek medical attention right away if they develop fever, headache, rash, or any other concerning symptoms.
Early diagnosis and treatment are essential for a good outcome.
Absolutely. Now, before we move on, I want to touch on this really interesting aspect of Rickettsia and their ability to manipulate the behavior of their arthropod hosts. It's almost like something out of a sci-fi movie.
It is fascinating, isn't it?
These tiny little bacteria can actually influence the actions of ticks, lice, or fleas. It's mind-boggling.
I know, and the mechanisms behind this manipulation are still being unraveled. But there's growing evidence that Rickettsia can alter the feeding behavior, the activity levels, even the lifespan of their vectors, potentially increasing their chances of transmission.
It's like they're hijacking those vectors, turning them into little transmission machines.
Exactly. For example, some studies have shown that ticks infected with certain species of Rickettsia are actually more aggressive feeders. They're more likely to bite and attach to a host.
It's like they're enhancing their own spread.
Exactly. And this manipulation can have significant implications for the epidemiology of rickettsial diseases.
It's a reminder that the microbial world is full of surprises. We're still uncovering those intricate ways that these organisms can influence our lives.
Absolutely. And this leads us to a broader public health perspective. Rickettsial diseases, even though they can be quite serious, they're often under-recognized and underreported.
So we need to raise awareness.
We do, both among healthcare providers and the general public. Clinicians need to have a high index of suspicion for these infections, especially during tick season or in areas where these diseases are endemic.
And public health agencies need to step up their surveillance efforts, their education programs, and their vector control measures.
Exactly. It's a team effort. By working together, we can raise awareness, improve diagnosis, and implement those effective prevention strategies to reduce the burden of these diseases.
It's all about protecting public health.
Yeah.
Now, I think it's time to shift gears again and talk about a topic that's really near and dear to our hearts as microbiologists. Horizontal gene transfer. Welcome back to the final part of our deep dive into Rickettsia. We've covered a lot of ground, from their unique intracellular lifestyle to the challenges of diagnosis and the latest in treatment and prevention strategies. But the world of microbiology is always full of surprises, isn't it?
It certainly is, and Rickettsia is no exception. There's always more to learn and explore.
And one area that I find particularly fascinating is horizontal gene transfer. It's this remarkable process that allows bacteria to essentially acquire genetic material from other bacteria, even those that aren't closely related.
Yeah, it's like they're swapping genetic blueprints, sharing those tools and traits that can give them an edge in survival.
And for Rickettsia, this ability to acquire new genes can have really significant implications, right?
Oh, absolutely. Horizontal gene transfer can help explain how these bacteria have adapted to different hosts, different environments. It might even play a role in the development of antibiotic resistance.
So it's not just about those core genes that define a species. It's about this constant acquisition of new genetic material that allows them to evolve and adapt.
Exactly. They're constantly fine-tuning their genetic repertoire to meet the challenges of their environment. And this can lead to new virulence factors, antibiotic resistance genes, even metabolic pathways that allow them to thrive in different ecological niches.
It's like they're always one step ahead, which makes studying their genetics and their evolutionary history all the more crucial.
It does. This knowledge can help us predict how they might respond to changes in their environment, like the introduction of a new antibiotic or a shift in climate.
It can even help us anticipate the emergence of new rickettsial diseases. Now, another area that's really shedding light on the complexities of Rickettsia is the research on the host immune response. We touched on this earlier, but it's such a complex interplay between our immune system and these bacterial invaders.
It is. It's not just a simple matter of our immune system attacking the bacteria. It's about finding that balance between clearing the infection and minimizing damage to our own tissues.
It's a delicate dance.
It really is. And we're learning more and more about the specific roles of those different immune cells, the signaling pathways involved, and the factors that ultimately determine whether the immune response is protective or whether it actually contributes to the disease process.
And I know there's a lot of interest in the role of T cells in controlling rickettsial infections. T cells are those white blood cells that can directly kill infected cells.
Right. And research suggests that certain types of T cells, particularly CD8 plus T cells, are really crucial for clearing Rickettsia from those infected cells. They can recognize those infected cells and eliminate them before the bacteria can spread further.
It's like having a specialized hit squad.
Exactly. And understanding how these T cells work, how they recognize and kill infected cells, could lead to new immunotherapies or vaccines that can enhance this protective immune response.
It's about harnessing the power of the immune system to fight these infections more effectively. Now, I know antibodies also play a role in controlling Rickettsia.
Oh, definitely. Antibodies are those Y-shaped proteins that are produced by B cells, and they can bind to very specific targets.
In this case, those sneaky Rickettsia.
Right. And antibodies can do a couple of things. They can neutralize those bacteria, preventing them from entering new host cells, and they can also tag them for destruction by other immune cells.
It's like putting a target on their backs.
Exactly. And researchers are now looking at ways to develop what we call monoclonal antibodies. These are very specific antibodies that are produced in the lab, and they could potentially be used as a treatment for those severe rickettsial infections.
It's like having a targeted weapon that can zero in on those bacteria without harming healthy cells. Now, I want to circle back to something we talked about earlier, something that I find both fascinating and a little bit unsettling.
Oh, you mean the way that Rickettsia can manipulate the behavior of their arthropod hosts?
Exactly. It's like mind control.
It's pretty amazing, isn't it?
These tiny bacteria can actually influence the actions of ticks, lice, and fleas, increasing their chances of being transmitted to a new host.
Right. They're essentially hijacking your vectors, turning them into more effective transmission vehicles.
So, for example, we were talking about how ticks infected with certain Rickettsia species become more aggressive feeders.
Exactly. They're more likely to bite and attach to a host.
And this manipulation can have a real impact on the epidemiology of these diseases, right?
Absolutely. It can increase the rate of transmission. It can lead to wider spread of those infections.
It's a reminder that the microbial world is full of these complex relationships, these intricate interactions that we're still trying to fully understand.
And these relationships can have significant implications for human health.
Absolutely.
Yeah.
Well, I think we've covered a lot of ground in this deep dive.
We have.
But as we wrap up, I want to leave our listeners with a few key takeaway messages.
Okay, sounds good.
First and foremost, Rickettsia species are fascinating. They're diverse, they have this unique intracellular lifestyle, and they can cause a whole spectrum of human diseases. We've got everything from mild flu-like illnesses to those really severe life-threatening infections.
Right, so it's important for clinicians to have a high index of suspicion, especially during tick season.
Absolutely, and early diagnosis and prompt treatment with antibiotics are essential.
Doxycycline is usually our go-to.
Right. And of course, prevention is key.
Always.
That means protecting yourself from tick bites, using insect repellent, wearing appropriate clothing, doing those tick checks. And it also means taking steps to control those tick populations in your environment.
And if you do find a tick, remove it promptly and correctly. Don't wait.
And remember, our understanding of Rickettsia is constantly evolving as new discoveries emerge. So stay curious, stay informed, and keep exploring this amazing microbial world.
It's a world full of wonders and challenges, and the more we learn, the better equipped we'll be to protect human health.